All right, translation. Okay, so last time we did a bit of an overview. So where does translation occur? Shouldn't say sometimes. Okay, so translation occurs on ribosomes, right? And that happens in the cytoplasm. They can be free floating ribosomes, or often they're attached to ER, specifically rough ER. So you should probably remember what ER stands for too. Oh gosh, I'm so mean for making you remember all this stuff. Ugh. Okay. All right. So we know that so far. And then the goal, of course, of translation. So remember the sort of overarching purpose of transcription is to use DNA as a template to make RNA. Okay. Now we're using RNA as the template to make protein. Good. Okay. We'll come back to this in a second. Here's the deal with translation. It's a bit more complicated. Okay. And the reason it's more complicated is because it involves more parts. There's more like players in order to make this happen. Okay. And so we're going to talk about all of those players and then we'll talk about the process. Okay. So actually, I want to do this a little bit different order than I have it listed in the PowerPoint. Okay. So one player, one part that we need is our, what is this? What do you think this is? What's this? Oh, man. I think I know how to use this by now. Right. So this is our message, right? This is our messenger RNA or mRNA. And this is, right, this is the template that we're using, right, to actually code for our protein. Okay. So, oh, look, there's a cap and there's a tail and there's the single stranded thing. It's been processed. Okay. I don't know if I'm going to sneeze or yawn or both. It's too early in the day for me to be yawning. All right. Okay. So you know this already. Okay. Here's another required entity. This is our ribosome. Okay. So before, when we were in the cell chapter, we were talking about ribosomes, we were looking at little teeny tiny dots. And it's like, that's a ribosome, that teeny tiny dot. Ribosomes actually have multiple pieces. They have different subunits. I don't care if you know anything about the details of those subunits. Okay. Quite frankly, I don't even really care if like you know the name A site and P site. Okay. What I really want you to know though is that like this is the place where the actual translating is happening. Okay. So this is what holds all the parts together. It's kind of the punchline, right? So there's this little slot. So see where it says mRNA binding site? This is the little slot where the mRNA hangs out. And then there are also tRNA binding sites. What the crap is tRNA? That's the other thing that we need. Okay. So far, in order for translation to occur, the different parts that are involved, we need the mRNA strand because that holds the message, hence the name messenger RNA. We need the ribosome because the ribosome is what holds all the other parts together. And then the last thing we need these molecules called tRNA. So tRNA stands for transfer RNA. Or if you want to, you can think of it as translating RNA because that's what it's doing. It's translating. So that's okay. It is a type of RNA. Okay. So much like the mRNA that we made, it has the same characteristics in the sense that it's made of RNA nucleotides, which mean that it can't have any T's for the bases, right? It has to have G, C, A, and U, right? Because that's what RNA has. It doesn't have T's. Okay. So what you're looking at, and these images are not in your textbook, right? What you're looking at is you're looking at like a diagram of a somewhat more realistic shape of what tRNA actually looks like. And then this is kind of like a simplified version to help you, look at images of this happening. Okay. Now, the thing about tRNAs is that you have a bunch of different tRNAs. Lots of them. Right? As it turns out, you have 61 different ones. Don't worry about that, though. You don't have to memorize that. Okay. So you have lots of different tRNAs. And what makes them different is this part here and this part here. Okay. All right. We'll come back to that. Okay. The last thing you need to know before we jump into all of this crazy mess is you need to know about something called the genetic code. So this is one of my favorite invisible biology topics because I just find the idea so enchanting that no matter what kind of organism you are, you're a human, you're a ladybug, you are a grass plant, you are bacterium, you are a brain-eating amoeba, whatever you are, right? All living things on Earth that we're aware of, right, share the same genetic code, okay? What the genetic code is essentially, it's like, it essentially is like a translation dictionary, right? So if you ever took a foreign language, I don't even know if people do this anymore because things have changed so much with school from when I was a kid. When I was in high school, we had to walk through the snow uphill both ways. Anyway, right? So when I was in high school and I was taking language classes, foreign language classes, one of the things that you had with you all the time was your little dictionary, right? So your English to Spanish and then Spanish to English dictionary, right? Or I took Latin in high school because I'm that kind of a nerd, right? So English to Latin, Latin to English dictionary, okay? Right? So whatever that is, okay? Essentially, the genetic code is that, right? So the way that they represent it in your book is in a circular form, which is cool from the perspective of finding what you're looking for, but I think is actually a little bit confusing just because it's like, it's a circle. So we'll come back to this in a sec. Actually, no, we'll start with this and then we'll start. Let's start. I don't know which one to start with. I find this one easier to understand, but this one is easier to use, like if you were trying to actively use it. Okay, so here's the deal about the genetic code. The way that RNA is used to code for a protein, right, is that there are 3-letter words, notice the air quotes, in DNA and in RNA that we call codons. Okay, so a codon three-letter word in RNA that translate to a specific amino acid. Oh, snap. What's an amino acid? Okay, so once again, let's go back to macromolecules, right? So proteins are these large polymers that are made up of individual units called amino acids, right? So amino acids are the individual monomers that build a polypeptide, right? Which is a strand of amino acids, essentially. Okay. All right. So codons are 3-letter words in RNA that code for, that translate to, right? That code for a specific amino acid. Okay. This little table shows all of the different possible combinations. Okay. So these letters should look familiar, right? U's and C's and A's and G's. Those are the bases that are found on the nucleotides, right? Now, why don't we see any T's on this list? Why isn't it like TCT? Why don't we see any T's? Because we're talking about RNA, right? So remember, RNA doesn't have T's. RNA has U's instead. So quite literally, what this table includes is it includes every single, I'm just underlining random ones, every single letter combination that can get you let me start over, sorry. Every single letter combination that you can have with 3 letters, and there's 4 letters to choose from. So every single 3-letter combination, given that we start off with 4 letters. I did not say that very well. I apologize. It works out to 64 though. Okay. So this table I like because it like lists them all and you can kind of see them and, you know, yeah. This table is a different way of presenting the same information. So this one is useful for if you were looking up something. So let's say I'm going to give you a random codon. I'm going to say CGA. I did not pick one on purpose. Okay. And I said, what's the amino acid that this codes for? This table is more useful for that because it tells you where to start. So, I start here and then I find, okay, my first letter is C. So I pick C. And then my second letter is G. So I pick G, right? So I'm working my way out. And then A is arginine, Arg. Okay, so this one is easier to use in that sense because let's say I asked you to look up CGA in the other one. You'd be like, okay, where are the C's? All right, well, the C's are all in this row here, and this is CU, and that's CC, and that's CA, CGA. Okay, arginine. So the contents of these 2 tables is exactly the same as far as like what it tells you, right? The difference is just how it's presented, okay? So in some ways, I find this one easier to look at. In other ways, this one is easier to use, okay? Regardless, I will absolutely never expect you to memorize this entire table. That's the kind of crap that they make you do when you take an upper division biochemistry class. I remember being like, really? I have to memorize this? Why? Why would I need to when I always have a little table that I can use for this? Like, this is ridiculous. This is a waste of neurons. Okay. There are a couple, though, that I do want you to remember, and those ones are highlighted. Okay. So these codons that I'm circling here, okay, are important because they play a role in the translation process, okay, that is significant. All right. So you will notice that every single codon that's here codes for an amino acid. Sometimes multiple codons code for the same amino acid. For example, everything that starts with UC codes for serine. It doesn't matter what the last letter is, right? UUU and UUC both code for phenylalanine. Okay, so it's redundant, the genetic code, right? Meaning that, right, sometimes multiple codons code for the same amino acid, but it is not ambiguous because each codon always codes for the same amino acid. Okay, so let's talk about those special ones for a second. So the special ones, The first special one shown in green here is called the start codon. Guess why the start codon is important? Because the start codon is where translation always starts. Translation cannot begin anywhere else except the start codon. Okay, so that's important. Now, as it turns out, the start codon also codes for an amino acid called methionine, right? So that's methionine or MET. By the way, you don't need to know all the amino acid names either, because who cares? You don't need to know that. You don't even need to memorize the abbreviations. You just need to know that each amino acid has a 3-letter abbreviation, and you need to know how to use the table. But you don't need to like, oh, I need to know the abbreviation for tryptophan. Like, no, we don't care. We do not care about that. Okay. All right. Where was I? I was saying something. Oh, the start codon. Okay. So the start codon always happens to code for methionine, but you don't need to memorize that because it's in this table. Okay. Which codon is the start codon? The start codon is AUG. This is super easy for me to memorize. You know why? My birthday, I was born in August, and you may know that the abbreviation for August is AUG. So I started in AUG, therefore translation starts in AUG. Okay? All right, you can remember it that way. Okay, so that was the start codon. Okay, now the stop codons. Don't try to memorize these because I don't need you to, fill up your brain with that. The only reason I expect you to remember start is because it's easy to remember, right? And now you know. Or if you don't want to remember it's my birthday, it's when the school year starts is August. Okay, so the start of the school year starts in August. Therefore, AUG is the start codon. Okay, which sucks, by the way. I hate having a birthday that's at the beginning of the school year. It's such a bummer. It's like, oh, we have to go back and what? Anyway, stop codons. Okay, so the stop codons, there are 3 of them. You don't need to memorize them. Happens to be UAA, UAG, and UGA, but don't memorize that because you always have the table you can look up. Okay. The deal with the stop codons is that the stop codons do not have a tRNA that matches up with it. So since they don't ultimately lead us to an amino acid, the whole process stops. This will make more sense when we get into the actual process. Okay. So for now, just they stop. They make it stop. Okay. And we'll talk about exactly why they make it stop in just a second. Okay. 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 I keep saying K. So codons are a 3-letter word in the RNA that codes for, that translates to a specific amino acid. Okay, cool. tRNA. So the tRNA, the transfer RNA, is the molecule that actually does this. Okay, so the one end is where those specific amino acids attach. So the amino acid hangs out on this end. This other end is something called an anticodon. God damn it, that's confusing, right? Dang it, that's confusing. Right? So what is that? What is an anticodon? Well, if a codon is what's written in the mRNA, the anticodon is going to be what matches it. Okay? So which example did we use here? So we said CGA. That was the example that I thought would be fun to do. So CG, and you can't see my red pen here, whatever. CGA gets us to arginine, okay? So that's the RNA. That's what the mRNA says, okay? This is the amino acid, right? So this is the codon, right, in the mRNA. So what would be the anticodon of that? Well, think about base pairing, right? Back to our base pairing rules. If our base pairing rules are A's and U's go together, because remember, we're not doing DNA, so it's not T's. A's and U's go together, and C's and G's go together, right? Then the anticodon, the sequence that would go with that would be GCU. Okay, so let's make this tRNA, right? Let's make it carry arginine. Okay, why not, right? So if the mRNA says CGA and that codes for arginine, right? I'm going the wrong direction. Okay, so I'm going to erase this just for the make space. Okay, so arginine is carried on that end. And then what is the anticodon going to be? Okay, so if our codon was CGA, then our anticodon is going to be GCU. Because then the RNA that matches it is going to be Does that make sense? I hope so. Okay, so the anticodon is what matches up with the codon. And what these tables are useful for is telling you, right, if you have this codon in the mRNA, this is the amino acid you're going to get. Okay. All right. Boom. Okay, let's talk about how that whole thing happens. Okay. All right. So once again, initiation, elongation, termination. There's, steps. And I'm not as concerned about you knowing the names of the steps as I am concerned that you sort of understand what's happening. Okay. So it turns out the ribosome doesn't all coalesce all at once. Okay. So here we have our start codon. What is the start codon? Same as the beginning of the school year, right? So start is AUG. So that sequence is where all of the components come together. Okay, so that little sequence, that codon, right, lines up with the ribosome. Then the very first tRNA pops in there. This tRNA is the one that has the anticodon that matches up with the codon AUG. And so because you know your base pairing rules, AUG is going to match up with UAC. Okay. And then because if we go back and look at our little table, right? AUG gives us methionine. There it is. Okay. And then the other piece of the ribosome hops on. So that's how this all comes together. Okay. All right. By the way, this is called the P site because this is the side where the polypeptide is going to grow. This is called the A site because this is where new amino acids come in. You don't need to memorize that. I'm just telling you if it helps you, if knowing why things are named what they're named helps you, cool. But you, like I said, you don't even need to know P site and A site. We don't care. Oh God. Okay. So this is not a picture in your book, right? This is sort of generic from another book. Oh God. Here's a more realistic image, but it comes with a little one that's the same, right? So the ribosome is brown instead of blue. Other than that, it's the same idea though, right? So there's our little start codon, right? Here's our little tRNA, and it's holding an amino acid. And the anticodon right here matches up with our codon. Oh, look. And there it is, right? So there's our RNA strand, our mRNA. And there's our ribosome, and there's our tRNA, and there's the little amino acid at the tip of it. Okay? Cool. Ugh. Here's how our polypeptide elongates, right? So it's kind of tricky. It's okay. We're going to go through slow. And remember, there are videos of all of this, okay? So here's our ribosome again, right? There's our mRNA. So we're kind of like imagining that we're jumping in here in the middle of translation happening, okay? So when this spot is open, the tRNA that has the correct anticodon that matches up with this codon is going to jump in there and sit in that little spot because the anticodon matches up with the codon. Okay? Then what's going to happen is this growing chain of amino acids called a polypeptide, right? A polypeptide is many amino acids linked together, is going to form a little bond here. You may recall that the bond between amino acids in a polypeptide is called a peptide bond. Okay, so that bonds to that. Now, this tRNA that was right here is now empty. So it gets kicked out. It falls off. It's empty. They can be recycled though. So it can be reused. It can get reloaded. Okay, so it goes somewhere else to get reloaded. Okay. Then now we have this one tRNA that has the growing chain and it gets scooted over to the next spot. Right? Well, actually what happens is the ribosome scoots down the mRNA, but whatever, right? So there's a scooting. So yeah, it stays attached to the mRNA and both the mRNA and the tRNA move relative to the ribosome. Okay? So then, and that's called translocation, but we don't care. Okay? So now this spot is open again. So what happened, right? Because this whole thing got scooted down. So now this is open again. So guess what? Now this is open again. Comes in, sits there. Then this binds to that. Now that's empty, so it gets kicked out. Everything scoots. Open space, we keep going. Until what? How does it know when to stop? It started at the start codon, remember? Whoa, not too far. Why are my children yelling so loud? I don't know if you can hear them. They're not murdering each other, I promise. They're supervised. Okay, so it started at the start codon. How would it stop? What would be responsible for stopping? That's right. The stop codon. Okay, so here's our, oh, my computer's getting laggy. Sorry. So here's that picture that we just kind of walked our way through. Here's a more realistic image. So there's our mRNA there. Here's that growing chain of polypeptides. Here's the new tRNA that's coming into that spot, right? And this little guy is like, boop, boop, transferring right there. And then this empty one's going to pop out, and then everything's going to scoot down. Yeah? How does it stop? How does it know when to stop? Let's look at the fake picture first, then we'll come to the realistic picture. Come on. Super slow computer. Okay. Oh God, there's a lot going on in this picture. Okay, so for now, don't look at any of this. Ignore that for a sec. Okay, right? So our mRNA comes out of the nucleus. This was all transcription, by the way, up in this corner, right? So the mRNA comes out. Here's it starting. There's the start codon. The thing logs on. The other thing comes in. They grow. Oh, that's how it stops. Because when you hit a stop codon, there is no matching tRNA. And because there is no matching tRNA, everything comes apart. Okay, so remember there were 3 stop codons. So all 3 of those have no tRNA that is complementary to them. They do not have a tRNA that matches up with them. Okay. So this is an image of all of that falling apart, right? So we hit a stop codon. There's no new, right? So, whoa, we hit a stop codon. There's no new tRNA that can come into there because there isn't one that matches it. There are no tRNAs that have an anticodon that match up with the 3 stop codons. Okay. So then the parts just, everything just comes apart. There's our protein. Well, polypeptide, because it might have some additional tweaking done to it. Our ribosome pieces fall apart. Our mRNA floats away. It can be reused. It can be translated again, potentially. Okay. And that little tRNA floats away. Everything falls apart. Okay. That's it. That's translation. Okay. Ugh. Harder to talk about. Okay. But in a nutshell, what's happening? tRNA carry amino acids to ribosome. Each tRNA carries an amino acid that matches, I should say, that goes with a specific codon. Okay. All right. So there is a lot more to this story, as it turns out, a lot more. We're not going to go there yet, though. We're going to save that. Okay. So later on in our next unit, right, we're going to talk about things like mutations, and we're going to talk about things like cell differentiation and like all of this stuff that's like the complicated bits. So this is just kind of like, this is the basic sort of entry level understanding of what's happening. And we're going to talk about all the weird stuff that happens later. Okay. So this is it for this section, for this unit. All right. And then we'll come back and do some of the more complicated and or crazy business later on. Okay. Watch the videos. I'm telling you, the little animations that I linked, and there's some in your homework too. There's all kinds of really useful tools on the internet for this. Okay. All right. Make sure you feel comfortable with this, because all the confusing stuff is going to make more sense if this makes sense. Okay. So get to work. Okay. Have a lovely one. Yes, I want to.